Good afternoon. My name is John Lane. I'm the superintendent of Wabash Conference and interim pastor here at the Bloomington Free Methodist Church. It's just good to see all of you here gathered this afternoon. I know we have several people connected with our church here, and we also have people connected with the Bloomington churches, the area churches, and we just bless you all, and we're so thankful for all the life-giving churches in the community. And we're glad that you're able to gather with us here today. I have the privilege of introducing to you Dr. J. Wile, who is a longtime friend of mine and someone who I have learned from over the years many, many things. We are all on a journey, aren't we, to try to understand this great world that God has created and our own existence, and trying to understand how learning and science and the church and the truth and the Bible all connect together. And Jay has been such a wonderful gift to the people of God and to those that are seeking the truth. And so, Jay, I want to thank you for all of the work you've done and all of the study you've done. Kathleen, thanks for putting up with them. Good job, because we all know who the real person behind all the scene is. So, as many of you know, Dr. J. Wile, he holds a PhD in nuclear chemistry and a BS in chemistry, both from University of Rochester. Assistant professor formerly at Indiana University and Ball State University. Currently an adjunct professor at Anderson University and Memorial College. He's the author of 14 textbooks, an award-winning teacher, internationally known speaker, and presents lectures on nuclear chemistry, Christian apologetics, homeschooling, creation versus evolution, several different, in many different countries. But I want to know, did you pick North Carolina to win? Probably not. All right, I don't know that hardly any of us picked North Carolina and Kansas. So today we're going to, at 2 o'clock, as we start here, the most exciting science related to creation. At 3 o'clock, we're going to have a break and have refreshments in the back. And then at 3:15, if you're able to stay, Jay is going to be talking about myth busting. And then at 4 o'clock, we're going to have a Q&A. And Dr. Jay really does love to interact. So if some of you, I know there's young people here who are using his books. And he'd love to have an opportunity to chat with you and hear what you're learning. And I know many of you have reached out to him through email, and you're always very good at responding in those ways as well. So let me pray for us, and then we'll welcome Dr. J. Wile. Our Father, we do just thank you for your grace, your goodness, your kindness. Lord, we thank you that you've launched us into this world, that you have crafted this place for us, that you're working toward a full redemption, and that you, Father, have given of yourself in your Son and in the Holy Spirit, that we may know who you are. Father, stretch our hearts, stretch our minds today. Help us to be able to understand a little bit more about what you have done, what you are continuing to do in this grand creation. Thank you for Dr. J. Wile. Thank you, Father, for all of the young scholars in the room. We just pray your blessing in their lives as they study and seek, Lord, to truly understand what you have done in this world in a greater and greater way. It's in the name of Christ we pray. Amen. So let's welcome Dr. J. Wile.
I guess I will go up there. Well, good afternoon. Thank you all for coming. So before I get started, I want to tell you my favorite thing to do is answer questions. So as I'm going through, and I'm breaking protocol a little bit, there's going to be some time for questions at the end of this talk and at the end of the next one. So we'll just, and the questions don't have to be on what I'm presenting. As I tell my wife all the time, I'm an expert on everything. She loves that. And actually, the 3 words that every scientist should be very comfortable saying is, I don't know. And I'm happy to say that if I can't answer your question for sure. But if you've ever had a question that relates to science and Christianity, I'm happy to take a stab at it at least. So what I want to talk about in this 5 or 6 hour session is the most exciting science related to creation. We're living in a time where a lot of scientific discoveries are being made, and they're pointing more and more strongly to the creationist view and less towards the evolutionist view. And so it's a really exciting time to be a creationist, in my opinion, because you see all sorts of stuff that's showing that. The biggest thing that's probably come across in the last probably 2 or 3 decades, really, is the latest news in genetics. When I was at university, I was taught that 98% of the human genome, 98% of all the DNA in your body was useless junk. That of all the DNA in your body, only 2% did something. Now, what they meant to say was, we only understand 2% of what's going on there. The other 98% we don't understand, so we just assume it's junk. And it fits really nicely into the evolution model that the genome would mostly be junk, because if you think what evolution is a trial and error process that, tries out something new. If it doesn't work, you die. If it does work, you're more likely to survive. And so you would expect in a trial and error basis, there would be a lot of junk. And that's what I was taught as scientific fact when I went to university. And from an evolutionary point of view, since the junk isn't doing anything, it's free to mutate and change as much as it's possible because it's not going to affect you, because it's not doing anything. But as it's changing and mutating and all that, it's possible something really good comes out of it. And that's where new genes are supposed to come from. The junk that doesn't do anything in DNA is free to try out new stuff and try to become some useful new gene. And that's the source of evolutionary innovation. Back when we started the process of trying to sequence the human genome to truly understand what's in our DNA, there were a lot of people who said, we shouldn't bother sequencing the whole thing because we know most of it's junk. So we should only bother to sequence the stuff we know works. So back in 1989, when the Human Genome Project was first started and people wanted to really study what this human DNA is, Sidney Brenner, who was a very important scientist in the UK, he said that it's only necessary to sequence 2% of the human genome, the part that contains coded information. The rest of the human genome is junk, so why bother to sequence it at all? Well, a couple things happened that fortunately changed that. One is it got a lot easier to do human DNA sequencing as technology improved. So what started out as a mammoth effort back in 1989 quickly became a lot easier to do. And so scientists thought, well, even though this 98% isn't really useful, we might as well sequence it because it's pretty easy to sequence. Other people thought, well, maybe that 98%, at least some of it, does something. So we probably ought to make sure we know everything that's in the genome if we're going to bother to do this endeavor. But even back in 2010, which is only, 12 years ago now, Dr. John C. Advice said this: the vast majority of human DNA exists not as functional gene regions of any sort, but instead consists of various classes of repetitive DNA sequences, including the decomposing corpses of deceased structural genes. And once again, this is exactly what you'd expect from evolution. As an organism evolves, some of its genes aren't going to be needed anymore. And so they'll eventually deteriorate. They'll eventually become the decomposing structures of deceased genes. So that's what was thought even just 12 years ago. There's a program called Avita. There are a lot of computer programs like this, but there's a program called Avita, which is considered sort of the gold standard of evolution simulations. It's considered to be the best working computer simulation we have for evolution. And it requires that 85% of all these simulated organisms the genomes of those simulated organisms start out as junk. So in order to produce evolution, the vast majority of the simulated DNA has to be junk, because once again, that's where the evolutionary innovation comes from. However, not too long ago, there was a huge effort. It was called ENCODE. And this effort involved the analysis of 1,640 different data sets for 147 different human cell types. You may not realize this, you've got a lot of different kinds of cells in your body. Your skin cells are different from your liver cells, different from your brain cells. In the end, throughout the course of your life, from conception to death, you have about 2,000 different types of cells in your body, at least at one time. As an adult, you have about 200 different types of cells in your body. So they looked at a good fraction of them, and they looked at a lot of data sets. And what they were looking for was how much of the DNA is actually used in these cells. As these cells go through the course of their life, how much DNA is actually used by them? And the result was 80%. As the journal Nature says, among the many important results, there is one that stands out above them all. More than 80% of the human genome's components have now been assigned at least one biochemical function. So this thing that I was taught as scientific fact when I was at university, 98% of the human genome is junk, is absolutely not true. At least 80% of it is functional and important. And in fact, if you talk to Ewan Burney, who is the project's lead data analysis coordinator, he says this: it's likely that 80% will go to 100%. Because remember, you've got about 2,000 different types of cells in your body from the time you're conceived to the time you die. They've looked at 147. So most likely there are other cell types that use other parts of the genome that we haven't seen the 147 use. So in the end, he thinks that all of the human genome is functional. It's used for something. As he says, we really don't have any large chunks of redundant DNA. This metaphor of junk isn't that useful. And once again, it was a scientific fact when I went to university. Now it's not that useful. One of the most interesting people to talk about this, John Stamatonoiopoulos, he's Greek. He was part of this team that did this project, ENCODE. And here's what he said. He said, I don't think anyone would have... Go back. I don't think anyone would have anticipated even close to the amount of sequence that ENCODE has uncovered that looks like it has functional importance. So he's saying this 80% number was a shock. Nobody would have anticipated it. But in fact, there was a whole group of scientists who actually predicted it. Not only anticipated it, but predicted it. And they were the creation scientists. The creation scientists have all said all along, the vast majority of all genome, whether it's human or some other organism, is functional. As a matter of fact, 10 years before the ENCODE projects announced their results, Dr. David DeWitt, who's a professor at Liberty University, made this video, and this video's title, if you can't read it, is Junk DNA is Not Junk. And so creationists have always said, look, if the genome is designed, and we think it's designed, then in the end, there's no way 98% of it is useless. There may be a useless thing here and there that has broken over the years, but just like, think about your car. Imagine someone designs a car, and then 98% of it doesn't work. It'd probably be called a Chevy Nova, right? But that car wouldn't run. Design structures aren't made to lose that much functionality. So, I've had a number of used cars over the years, and they've all had personality. So I had one car where the thing that you pull out for the headlights had broken off, so I had to use pliers to pull out the headlights. And the car that I just had before I got my last car, the wipers only worked on high. So no matter how much it was raining, for me it looked like a thunderstorm, right? So, a few things can break in a car, and the car's okay still. But if too many things break, the car just doesn't work. If DNA is designed, you'd think the same thing. A few things could break, but in the end, if too much stuff breaks, it's just not going to work anymore. So, if you believe that DNA is the result of design, you would not expect 98% of it to be junk. And that's what we find. Now, there are some evolutionists who are actually honest about what these implications are. And the one that I think is at least the most honest, he looks like a professor, doesn't he? His name's Dr. Dan Grauer, and he's a professor of biology at the University of Houston. And remember, this project that says at least 80% of the DNA is functional, that's called ENCODE. He says, if ENCODE is right, then evolution is wrong. And of course, for him, that means ENCODE can't be right. Because he's an evolutionist, it can't be right. So evolution can't be wrong, so ENCODE must be wrong instead. Because he recognizes that in order for a genome to evolve, the vast majority of it has to be junk. And if genomes aren't junk, they can't evolve. But in 2021, the Oxford Journal Genome Biology and Evolution stated, quote, the days of junk DNA are over. So junk DNA was a phrase that I heard throughout college, throughout grad school, reading papers in the scientific literature. But that whole idea is gone now because we know the genome is mostly functional. And once again, this is what creationists predicted and evolutionists didn't expect. The authors of this paper actually said the same thing. They learned as fact that DNA has lots of junks, but today's research shows it's just not true. So that's one thing I would say. Anybody who's at college right now, going on to university, even in high school, a lot of the things you learn as fact are going to be shown to be wrong. That's just the way science is. We develop ideas, we test them, but in the end, because our testing can be flawed, because our reasoning can be flawed, a lot of what you learn as fact today will not be fact in 10 or 20 years. And that's just the way science is. All right. So, consider what one leading evolutionist says about all of this. This guy, a very well-known evolutionist, Jerry Coyne, he wrote a book that's called Why Evolution is True. So you know his point of view going in. He says, for a theory to be considered scientific, it must be testable and make verifiable predictions. And if those predictions are met, it gives us more confidence that the theory is true. In fact, he's absolutely right here. Scientific theories don't explain things. I can make up any story to explain any piece of data. That's not what makes a theory scientific. What makes a theory scientific is that based on that theory, I can make a prediction about something not known, and when it's tested, that prediction turns out to be true. That's what makes a theory scientific. A theory that only explains things is like a bedtime story. A theory that can predict something that's testable, that's a scientific theory. Well, in fact, the creation theory has made many testable predictions, which have been confirmed by the data. And often these testable predictions are precisely opposite of what you would expect from evolution. So one that's really important to me is there should be significant amounts of carbon-14 in dinosaur bones. This is important to me. I'm a nuclear chemist, and nuclear chemists deal a lot with radioactivity. Carbon-14 is a radioactive isotope. You have carbon-14 in your body, and that carbon-14 is emitting radioactive particles right now to the person that you're sitting next to. In fact, for most people, their greatest exposure to radioactivity is sleeping with their spouse. Because you're close for a long time, and you and your spouse are both radioactive. You're both bathing each other in your radioactive particles, right? And what's worse for my wife is the more fat you have, the more radioactive you are. So I'm a big source of radiation because fat cells sequester potassium, and potassium has a radioactive isotope. So in the end, everything living is radioactive. Carbon-14 is in every living creature, so every living creature is radioactive. However, radioactive isotopes decay. Carbon-14 decays into nitrogen. So if I had a lump of carbon-14 in my hand right now, it would look like, a piece of charcoal, because it'd be black solid. But as it decayed away, it would disappear, because it would become nitrogen-14, which is a gas, and it would float away. So over time, if I have a bunch of carbon-14, it disappears. And unless it's replenished in some way, it continues to disappear. And carbon-14 cuts itself in half every 5,700 years. So if I had 100 grams of this carbon-14 and waited 5,700 years, roughly the length of this talk, then in the end, I would have 50 grams. If I waited another 5,700 years, I'd have 25 grams. So every 5,700 years, it cuts itself in half. Well, if you think about it, no matter how much I start with, if I keep cutting in half, after 10 or so times, you don't have much left. So the general rule of thumb is carbon-14 becomes undetectable after about 60,000 years. Because once an organism dies, it can't replenish its carbon-14. So the carbon-14 starts to decay away. And since there's no way to get it back, it just goes and goes away and goes away. After 60,000 years, it will have cut itself in half 10 times. That makes it so small, you can't detect it anymore. So that's the general rule of thumb. Well, dinosaur bones are supposed to be 65 million years old. So how much carbon-14 should be in them? None. You shouldn't be able to detect carbon-14 in anything less than 60,000 years old. In fact, every dinosaur bone that's ever been tested for carbon-14 has plenty of it in it, right? For a long time, evolutionists wouldn't even test, and evolutionists still don't, test dinosaur bones for carbon-14. The argument is, why would we bother to test for something that we know is not there? Because after 65 million years, it should all be completely gone. However, creationists said, no, these bones are not 65 million years old. They're thousands of years old. So if you analyze them, there's going to be carbon-14 in them. Well, sure enough, eventually creationists were able to raise enough money to find fossils of their own. This is actually a picture of a creation scientist cutting into the femur of a triceratops. And he's cutting into it, of course, because you don't want to test the outside of the bone. There's lots of contamination there. You want to test the inside of the bone, where there's still residual bone matrix from the dinosaur. And so in the end, he's cutting in. He's going to take a sample of that interior bone, going to send it off to a lab. This lab makes money by testing for carbon-14. And in fact, when they do that, they always find carbon-14 in the dinosaur bone. So for example, this Acrocanthosaurus fossil is supposed to be 100 million years old. However, the carbon-14 that's in it indicates it's only 23 to 30,000 years old. Now, I don't think it's 65 million years old or 100 million years old. I also don't think it's 30,000 years old. I think both of those dates are wrong. But the fact is, these two dates are utterly incompatible with each other. If this dinosaur bone really was 100 million years old, the carbon-14 test should come back, it's undateable. We can't find any carbon-14 in it, so we can't give you a date. But in fact, they give you a date of somewhere between 23 and 30,000 years old. This Triceratops is supposed to be 65 million years old. Carbon dates between 24 and 39,000 years old. So in the end, there's something wrong here. Either one or both of these dates are wrong, because they can't both be true, right? In fact, if we've, all of the ones that have been done so far, 8 dinosaur fossils from all Texas, Alaska, Colorado, Montana, China, and Europe, all had so much carbon-14 in them that the oldest any one of them could have possibly been was 39,000 years old. Not a single one of these samples tested as having 0 carbon-14 in them, which would be expected by evolutionists. So here we have a problem. We have dinosaur bones that are supposed to be millions of years old that are carbon dated to be only tens of thousands of years old. And the fact is, if I'm doing a carbon-14 date on some bone and I don't assume that it's millions of years old, and I get a number like that, I believe it. Because carbon-14 dating is supposed to work. So in the end, if I see some bird bone, some bird fossil, I carbon-14 test it, it turns out to be 39,000 years old, most scientists would believe it. However, they don't believe any of these numbers simply because they come from dinosaur bones. And dinosaur bones aren't supposed to be that old. Now, if you're someone who really believes these dinosaur bones are millions of years old, how do you deal with this? How do you deal with this kind of information? Well, here's how the scientists at this carbon-14 dating lab responded. They said they won't test any more samples sent to them by creationists. So here's the letter. This is part of a letter they sent to the creation scientists who were sending them samples. Now remember, this lab makes its money by dating these samples that people send to them. And the lab's name is CAIS. The scientists at CAIS and I are dismayed by the claims that you and your team have made with respect to the age of the Earth and the validity of biological evolution. Consequently, we are no longer able to provide radiocarbon services in support of your anti-scientific agenda. Now, that's a phrase that's gotten really popular over the last couple of years, hasn't it? Right? This was a few years before that. But I want to ask you, what's anti-scientific? Asking for more tests to be done, or refusing tests because you don't like the answers? Which is more? You've got an answer. What is it? Let's go ahead. It's refusing to do tests, right? That's what's anti-scientific. See, he knew, right? So that's a crazy thing. Let's suppose I really believe these dinosaur bones are millions of years old. I don't, but let's suppose I did. If I did, I would want to know why in the world there's carbon-14 in them. So what would I want to do? I'd want to do a lot more tests. I'd want to test a lot more bones. I'd want to see if there's, some that don't have carbon-14, some that do. What's the difference? That's the way you find a scientific answer. You do more testing. You don't do less testing. But that's what these folks did because they didn't like what their tests were showing. And that's truly unfortunate. Unfortunately, we're going to see this again. So, dinosaur fossils have carbon-14 in them. They shouldn't have carbon-14 in them. Here's something that's even more amazing. Dinosaur fossils have soft tissue in them. I don't know if you've heard about this. It's not widely trumpeted. It's well known in scientific circles, not widely trumpeted in the media. The first example of this comes from 2005. Mary Schweitzer and her colleagues were trying to excavate this Tyrannosaurus rex fossil. They ended up breaking the femur, which is the big bone that goes from the knee to the hip. And inside, they found tissue like this. And in her paper, she actually says, you grab this with forceps, you grab that with forceps, and you pull, and this little guy stretches. And when you release, it goes right back to this size. It's truly elastic. Now, for someone who knows enough biology, it's clear what this is. This is muscle tissue, and this is probably a ligament that connected the muscle tissue at some point. So anyway, this is muscle tissue, and it's soft after 65 million years. Now, we know that most of the time, soft tissue decays away in about 50 weeks. Now, sometimes it can be frozen. We found frozen mammoths, for example, that have some soft tissue left in them and so forth. So it can last longer than 50 weeks under the right conditions. But here's the key. This soft tissue is made up of molecules. And these are molecules we've studied pretty well. And based on our understanding of these molecules, the molecules themselves break down after about 30,000 years. We haven't tested this, obviously. We haven't waited around 30,000 years yet. But based on our knowledge of these molecules and based on the computer models that we use, these molecules themselves last only about 30,000 years. So after 30,000 years, the very molecules that make up this soft tissue should be gone. But they're not. They're still there. And in fact, the guy who basically is in charge of this whole fossil and this soft tissue and everything, he's a fairly well-known guy. I'm not sure if you've recognized the name, Jack Horner. He's one of these fossil hunters that kind of breaks the rules to get fossils and things like that. So he'll go into a country he's not supposed to go into secretly, and he'll go get fossils that he knows are there and so forth. So he's kind of this cowboy among the paleontologists, right? It's his museum that Mary Schweitzer was working for when she made this discovery. So it's his museum who owns the fossil. He was offered $10,000 for someone to do carbon-14 dating on the tissue. And he said he was not going to do this because the spin that creationists can get off it is not going to help us. So anyway, he doesn't want to know how much carbon-14 is in that sample because he figures there's some in there and he just doesn't even want to know, right? And in fact, this $10,000 grant was above and beyond the cost of the test. The people that were going to give him this grant, that $10,000 was after the test was done. And they sweetened the deal saying, look, we'll let you date 4 other artifacts with carbon-14 dating as well. So this is a sweet deal for the museum. And the head of the museum says, I'm not going to do this. And the reason? He knew he wouldn't like the answer. So the best way to not deal with the answer is to not do the test, right? So in the end, lots and lots of examples have been found of this. So for example, this is a Triceratops. This is the a skull of a Triceratops. It's not the one that I'm talking about. It's just an indication of what a Triceratops skull looks like. Some creationists had found a horn from one of these skulls. And the horn by itself isn't worth that much. So they decided to see if they could find any soft tissue in it. So what they did was they soaked it in a weak acid. If you haven't done this experiment yet, you really should. Take some vinegar, which is a weak acid, and put an egg in it and leave it overnight. In the end, the vinegar is going to eat off the entire shell, but it won't attack the tissue underneath. So you will have a bird's egg with no shell. And it is cool. It's really soft and squishy, and everything. Because weak acid tends to be able to react with minerals, but not with biological molecules. So in the end, they soaked this horn in weak acid for a few months. And in the end, when all the minerals were gone, there were strips of soft brown tissue. There's a documentary called Is Genesis History? Not sure if anybody's seen that. It's a really interesting documentary. But they have actually video of the guy who's got this tissue, and he just stretches it and releases it, and it bounces right back. It's this soft, stretchy tissue. Now, of course, this tissue could be anything. So they decided to look at it under the microscope. And guess what they found? They found bone cells. They found exactly what you expect to find in bone. Right? So this isn't some contamination that occurred after the fact. This is actually a sample of the Triceratops horns bone tissue that's still soft. And so in the end, they went under an electron microscope where you really see this well. There's a bone cell, that's another bone cell, that's another bone cell. Bone cells have a very characteristic shape. Most cells are either circular or spherical or mostly spherical. Bone cells are much more oval, and more importantly, they have these little hairs that stick out of them. They're not really hairs. They're called filopodial extensions. That's science for hairs. And these little filopodial extensions, they allow the bone cells to communicate with each other. Because bone cells have to be able to communicate with each other to figure out how to maintain the bone. So, for example, if you gain weight, your bones have to put on more mass. So what the cells, they talk to each other and they say, this guy's gaining weight, and it's making my job harder. So we're all going to have to make more bone to make it easier. So they have to communicate. They communicate by touching each other with these filopodial extensions. So when you see a shape like this, you know it's a bone cell. And in the scientific paper where this was published, the author says this: filopodial extensions, those little tiny things, are delicate and showed no evidence of permineralization or crystallization artifact, and therefore were interpreted to be soft. These are dinosaur bone cells that are still soft. They haven't been hardened by fossilization. They are still soft. Right? Now, they're not alive. They're dead. But they haven't hardened and they haven't decayed away. And if you look, this little black line going from here to here, that represents a length of 20 millionths of a meter. These little filopodial extensions are significantly smaller than that, and they haven't decayed away yet. And they haven't fossilized. That's astounding. That's really, really astounding. They carbon dated the horn. It turned out to be 41,000 years old. So once again, carbon-14 dating doesn't make sense related to the age of the supposed age of these fossils. Now, once again, you might wonder how evolutionists respond to this. This is the guy who wrote the paper. He got fired. Yeah, as a matter of fact, according to the lawsuit that he filed, at one point, one of the university officials stormed into his office after the paper was published and shouted, quote, "We are not going to tolerate your religion in this department," end quote. And of course, that turned out to be bad for the university. Now, I've spoken, this guy's Mark Armitage, and I've spoken to him on several occasions, really interesting fellow. He says he doesn't think it was the scientific paper that got him fired. In the end, what got him fired was once the scientific paper came out, students knew all they had to do was go to his lab to see dinosaur cells. And if you're interested in geology or paleontology, why wouldn't you want to see dinosaur cells? So he said basically every day, he would have 5 or 6 students throughout the course of the day come and say, "Can I see the dinosaur cells?" And of course, he'd show them. He'd love that. He loved that. He said that was the problem. Because, of course, these students were confused. about how in the world there could be dinosaur cells after 65 million years. He says he thinks that's what got him fired. Well, anyway, he ended up winning his lawsuit, and the amount was undisclosed. Who knows how much it was? But he's been using that money to fund his own further research on these soft dinosaur cells. He's actually isolated them completely away from the bone and has them floating in liquid. He's got a website, dstri.org, so the Dinosaur Soft Tissue Research Institute, DSTRI. He's got a video on there where he grabs one of these cells with microscopic forceps and flops it back and forth, and you can just see it flopping around. It's completely soft. Now, once again, it's dead, but it's soft. And you really honestly want me to believe that it's been hanging around for 65 million years and has stayed soft? That's not the way tissue works. That's not the way these chemicals work, all right? And it turns out this is incredibly common. Paleontologists specifically want to know, how common is this? Is this kind of a rarity or what? So they took poorly preserved dinosaur bones from a museum. They just went to it wasn't even dinosaur bones that were on display. They were in the back room. They were as one of the authors said, they were very scrappy, individual broken bones. I can't even tell you what dinosaur they came from. But in the end, when they looked for soft tissue, what did they find? They found red blood cells and they found proteins. Both stuff that shouldn't exist for more than 30,000 years. So in the end, dinosaur soft tissue is common. And here is the current record holder. The current record holder is this tube worm. He's supposed to be 550 million years old. And yet when the scientists looked inside him, what they found was soft tissue. They say it this way, minerals have not replicated in any part of the soft tissue, and the carbonaceous material of the wall is primary, preserving its original layering of the walls, its texture, and its fabrics. So what he's saying is, it hasn't decayed away, it hasn't fossilized, and it's supposed to be 550 million years old. So if you really want to believe these dates the paleontologists give you, you have to figure out some way to keep living tissue, or not dead tissue, soft for 550 million years. That seems ludicrous to me. And as a chemist, I cannot understand any kind of chemical mechanism that it would allow this to happen. And certainly, they haven't come up with one either. So in the end, that's a really significant find. And once again, this started in 2005, so it wasn't all that long ago. This particular paper about the 550 million-year-old worm was just published 8 years ago. So this is a real well-known thing in paleontology and science circles. It's not well known in the general public. Now, if you want to get people interested in science, don't you think you'd talk about stuff like this? Don't you think you'd say, hey, we've got soft tissue from dinosaurs hanging around. That sounds really interesting to me. But we don't talk about that. That's one of these problems that we're not allowed to talk about. But in fact, I think it's very, very significant. All right. Here's another creationist prediction. Remember, we're talking about predictions that creationists made that are testable that came true. So far, we have that junk DNA is not junk, carbon-14 in dinosaur bones, and soft tissue in dinosaur bones. Here's another one. And this is something you learned, and I'm sure you learned this as fact. If you look at your large intestine, near the entrance to the large intestine, there's this little worm-like sac that hangs out. It's called the vermiform appendix, and vermiform means worm-like. But most of the time we leave out the vermiform part because it just sounds weird, right? So this is your appendix, right? And it hangs off your large intestine, and you were probably taught this as scientific fact. We have an appendix, a small remnant of a prior ancestor species intestinal sac, which not only is of no use to us, but which can sometimes kill us when it gets clogged up and infected. What kind of god or other intelligent designer would design organisms with such useless, imperfect, wasteful, and sometimes even harmful physical features? This is from a book that was published in 2006. So even in 2006, books were saying the appendix, it's useless. It's actually a remnant from a prior ancestor. You all heard that? In fact, up until pretty recently, really up until about 10 years ago, it was considered routine that if you were doing an operation on someone to remove something else and you had access to the appendix, you just snipped it off. Even if it wasn't, even the person wouldn't have any problem, you just snipped it off. because it's useless and it could burst later, and that's bad. So just as a preventative measure, if you're in there, snip it off. That was really, really common. And that's because it was just considered scientific fact that this is useless. We now know that's not true. And once again, creationists were the first to predict this. Back in 1988, J. Warwick Lover said this: the appendix would be involved in the control of which essential bacteria come to reside in the cecum and colon in neonatal life. As well, it would be involved in the development of systemic tolerance to certain antigenic agents within the alimentary tract, whether they are derived from bacteria, foodstuffs, or even the body's own proteolytic enzymes. So getting rid of all the $20 words, what he's saying here is that appendix is really important for maintaining the right bacteria in your colon and fighting off pathogenic bacteria. Turns out we know that's exactly right. All right? So a study of people with recurring Clostridium difficile infections. So these are infections that occur in the large intestine. Clostridium difficile is a bacterium, and it causes all sorts of intestinal problems and so forth. You can take an antibiotic and get rid of them, but they tend to come back. Even if you take all the antibiotics, just as the doctor says, it'll clear up for a while, but it'll tend to come back. So that's what recurring means. So you have it, you get over it, it comes back, you get over it, it comes back, keeps coming back. We now know this happens mostly in people without an appendix. If you have an appendix, you are very unlikely to have these Clostridium difficile infections, recurring infections. And the authors specifically say it's because with those with an appendix can recover their normal bacteria sooner. Now, I think the reason a lot of people thought the appendix was useless was because in the U.S., you can snip it off and nobody has a problem, right? However, the first indications that that wasn't true was when people started doing appendectomies in third world countries. And they found that these people then became more likely to die of intestinal diseases. Then this study comes out in the U.S. saying, even in the U.S., people without appendix tend to have this reoccurring infection. So now we know what the appendix does. This appendix is basically a hideout for the good bacteria that live in your intestine. So you have all these wonderful bacteria that you provide food and housing for, and in exchange, they provide you with chemicals that you can't make yourself and other things that you need. And as long as everything's working fine, these bacteria are living fine, you're living fine. If you get an intestinal infection like that, those bacteria tend to get killed by the infecting bacteria. All right? What happens is, when that's happening and you have an appendix, the good bacteria hide out in the appendix. And if you look at the appendix, it's actually designed like a fork, or like a fort. There are 3 rings of lymphoid tissue that fights anybody else trying to come in. So the good bacteria are allowed in, and then those little rings of tissue fight off any bad bacteria that try to get in. So basically, the appendix is a hideout. Once those bad bacteria are gone, the good bacteria come back in. And so it basically helps you maintain the bacterial balance in your colon. Really quite incredible. But once again, creationists always said the appendix has a function. Evolutionists said no, it's a useless remnant. We now know the creationists were right. All right, that's what I've got for this talk. Oh, we got questions already? Go ahead. Oh, well, let's take questions first. Would that be all right? So, any questions? Questions about or questions you want a scientist to answer? Go ahead back there, yeah.
I'm 75 years old. And when I was in high school, I took a science class that had a science teacher that emphasized questions and looking at the data. And it seems like today that people who ask questions in our current woke culture are shut up and told you're all about hate and disinformation, and this is supposed to be the science community. And I'm even getting very distrustful of the medical profession because they seem to be part of the you don't ask questions. If I go to my doctor and ask a question, since I'm in a political arena and pretty much told, you don't know anything, shut up, you can't ask questions. And then if I present any information I've had, no, that's hate speech and that's misinformation. I'm wondering if the scientific community is no longer scientific, wanting questions and looking at the data and wanting truth, but has joined a political, cultural group of the one world government and leading to the Antichrist, and they are all against truth. Truth must be banned, and all they are for is against God.
Well, I'll tell you this. It's really fascinating if you look at the history of science, that, initially we had thinkers like Aristotle, and no one would question his conclusions. And so Aristotle's around in 300 BC, and even by 1500 AD, you weren't allowed to question Aristotle seriously, because Aristotle was the expert, and you don't question the expert. What caused the scientific revolution were two guys, Copernicus and Vesalius, who both questioned the expert. Copernicus said, no, Aristotle's wrong, the earth isn't at the center, the sun is. And Vesalius said, no, Aristotle's wrong, almost everything he taught about human anatomy is false. And the two books that were written in 1543 are thought to have started the scientific revolution because we started questioning authority. That's what turned us into real scientists. And over the years then, it became very important to keep questioning everything. Now we seem to have gone backwards. And we saw it first, I think, with creation evolution more than anything else. If you didn't like evolution, you weren't supposed to question it. And if you said things against evolution, then you were crazy and you didn't get your university position and all that kind of stuff. But now, even with this COVID stuff, if you question anything Anthony Fauci says, you're anti-science. How is it anti-science to question? And I do take that point very seriously. It seems like we've taken a big step backwards in terms of how we deal with science. Now, I agree, Anthony Fauci has forgotten more medicine than I know. So in the end, he's a good resource to listen to. There's no question about that. But you don't just blindly trust someone because he or she is an expert. You've got to look at the data yourself. Real early on this pandemic, I was given a speech at a commencement. It was held outside, and all these, spacing restrictions and everything. And I said back then, we're in the middle of this pandemic, and let me assure you, as a scientist, no one has any idea what we should be doing. So look at all the information you get, try and figure out what's best for you, do that, and please show grace to other people who do things differently. And that's the way Christians should have approached this pandemic. Yeah, there are lots of sources of information, and all of them may be spouting some truths. But in the end, you've got to figure out what's best for you, what makes sense for you. But then please, if someone else chooses some other path, don't condemn them for that. In the end, they're just looking at different information. And even now, 2 years into this, I can still say, for sure, there's an enormous amount of stuff about this virus we do not understand at all. And to just not question it, that's wrong. That's not science. Go ahead.
I looked into this research, and it said they're working on self-assembling nanobots.
Yeah, self-assembling nanobots.
They made organs, like they can put it like use proteins, and they can literally inject them into your body, and they can go and do function. Like they use nanobots to go target cancer cells, and some.
Yeah, there's some experimental research that's being done on that, specifically on animals, where these proteins, they call them nanobots. That gives a sort of a fake, they're nano size, so they're very small, but they're proteins that are doing what proteins do. It's not like it's some robotic thing. They've designed the proteins to do what they're doing.
So started in NASA. It started as they were trying to go from copper cores. They didn't want to try to get rid of it because all weight it makes the amount of money. So they're supposed to be working on efficiency, but to lower the cost. And they've been working on nano stuff way longer than they let on. And they have started shrinking it, and they've added proteins and carbons to it. They are circuit, like little
Yeah, but that's not happening like. I mean, a lot of this is really theoretical, and what's happening is mostly happening in animal models, the ones that are doing this. But it's a little different from that. It's not. There are ways of trying to treat cancer, for example, using these proteins. But these proteins are actually building complexes that fight the tumor. And this is, none of this has been approved for people yet. So that's a long way off for people if it happens. So, yeah, if you want to show it to me afterwards, that'd be great. Yeah. Anything else? Any other questions? Yes, ma'am.
So you mentioned, and we all know that creation scientists have a rough time today, and they have a rough time studying and continuing their research as well as everything they take from the outside world. But what's your advice for young people who are interested in going into a science field where they're most likely to face having to go through a PhD program or something where they're going to be taught evolution? I don't know of any programs that are PhD creationist studies. So what's your advice for people that want to go down that path?
Oh, that's a great question. So, if you're interested in science, and you want to go pretty far in it, you are going to encounter a lot of evolutionary dogma and doctrine. There's no way around that. I think part of it depends on your personality, whether that's something you can do or not. I personally thrive on being challenged. I remember I was answering this email. We were on a plane. My daughter and I were on a plane, and I was answering this email, and I was obviously just typing very animatedly. And she said, "What are you doing?" And so I explained this little argument I was in with this other scientist. And she looked at me, she goes, "You live for this kind of stuff, don't you?" So I think when you have that kind of personality, that can be a very good thing. And there are a lot of very well-known creation scientists today who got PhDs in evolutionary universities. Kurt Wise is a creationist paleontologist. He got his degree under Stephen Jay Gould, who at the time was the most prominent evolutionary evangelist that existed, right? So when you have that personality, that energizes you. And I do think if you want to challenge evolution, you're going to have to learn it. And you're not going to learn it from a creationist telling you what evolutionists believe. That's not how you learn it. You have to learn it from evolutionists telling you what they really believe. That's how you do it. So I think it takes a particular kind of personality to do all that. But I think those are the most effective anti-evolution people, or the people who studied it. Or the other one, which is not your question, but the other one is the people who started doing research, and their research showed them evolution doesn't work. One of the greatest examples of that is Dr. John Sanford. He's a geneticist who his own research said this doesn't work, and so he became a creationist, right? So those are the best evolutionary, or the best creationists, because they understand what they're fighting, right? So if you don't have that kind of personality, you can still do science, but it needs to be more applicable, because evolution is so useless from a standpoint of what makes a theory useful. That in the end, if you go into medicine, you'll have to bow at the altar of Darwin a few times, but that'll be it. Because in the end, there's nothing really that evolutionists gives you that makes you a better doctor. So I think if you go to the more applied biological fields, or if you go to chemistry and physics, then those kind of fields, you don't encounter that at all. But yeah, it's a tough thing. And as someone growing up, I got my PhD I was a Christian. I got my PhD in a very evolutionary bent school, but I was a chemist, so we didn't really deal with that. But as I was, being a professor and so forth, you could see how my creationist leanings were causing me to lose, credibility within my own institution and things like that. I was eventually denied the opportunity for tenure, I think, because I was a creationist. So, I think that happens, but what I believe and what my wife helped me see way back when was that, if God's putting you on this path, He's going to find some way to make this path work for you, and it may just not be the traditional way. And that's when I started writing test books and things like that. So, does that answer your question? Yeah. And one other thing I'd say for you aspiring students out there who are going to go to university, when you're in a class with a professor, you have to give the professor the respect he or she deserves, regardless of whether you believe what he or she is teaching you, because that's something you do. You give respect to people who are in authority over you. At the same time, you don't give up your beliefs just because they're preaching to you, right? And so, for example, the balance I found, and this works for me because of my personality, the balance I found was when I was in a biology class or any other class where I had to answer a question related to evolution, I would always give the evolutionist answer, then I'd put a little star. And at the bottom of the paper, I'd say, this isn't the correct answer, but it is the one you're looking for. And that's how I showed that I understood the material, but I just don't agree. Right? So, and definitely don't try and argue with your professor in class. Right now, I could take the evolutionary position and wipe any one of you out in a creation evolution debate. I just know more. So, don't try and argue with your professor about that. Other questions? Yes, sir.
This is kind of pretty general, but it always troubles me. I want your comment on the caveman versus Adam and Eve.
Oh, the caveman versus Adam and Eve. Well, what's interesting is the caveman concept is really fallen out of fashion a lot in anthropological circles, because in the end, what we found is that even these people who lived in caves had incredible social structures, they had jewelry, they had art, they had everything you'd think of as being people. Right? So people who lived in caves were probably just people. And they lived in caves because it was convenient, it was, regulated temperature and things like that. They lived in caves because it was convenient, not because they couldn't do something else. What we do know is if I look at the fossils that we see that are supposed to relate to human evolution, what I see is a lot of ape-like fossils. And then I see a lot of human fossils, and I don't see anything in between. And the few things they try and put in between seem really forced to me. So, for example, if you know what Lucy is, Lucy is this famous fossil, supposed to be one of the steps between ape and human. You put every one of her bones in a computer, and the computer says unequivocally, this is an ape. But because it's possible for her leg bone to connect to her foot bone, through her ankle bone, in such a way that she could have stood up straight comfortably, that makes her part of the human line. Even though every bone's an ape bone. And that's what I see. There's a really famous comment made by Esteban, a paleontologist. He said, you look at all these fossils, and they all come from Africa, and they're all supposed to be part of the human evolutionary line. You know what you never find in Africa is the fossil of an ape. You only find the fossil of something in the human line. How is it there are all these apes in Africa and we never find any of their fossils? Because in the end, what he's saying is you guys are all over-interpreting this. So, yes, people lived in caves, and as we've learned more, they have an incredible art and culture and everything else. And then we have a lot of apes. So I don't see a lot of... and if you look at these drawings of, that supposedly show the human evolutionary line, please understand these drawings, usually they're put out of order. So, they're shown as, ape-like, less ape-like, less ape-like, man-like. But in fact, if you ask the paleontologist, where do these bones come from? Well, this guy really belongs over here, but he looks better if he's over here. So almost every one of these drawings you see of the human evolutionary line, first of all, way over-interpreted, and secondly, usually out of order. So, yes, sir?
I'm glad you got on from Alex Lillian.
Hi.
I might have misunderstood you, but did you say carbon-14 dating doesn't line up with evolution, but it also doesn't fully line up with our view of how the age of the Earth?
Right, yeah. No, and that's where we are with carbon-14. The problem is all of these radioactive dating methods have assumptions built into them. And the quality of the method is only as good as the assumptions that are in it. And the problem is, basically, if I look at any one of these methods and look at one of their assumptions, I can usually find a problem with it. So, for example, in carbon-14 dating, we have to figure out how much carbon-14 was in the organism when it died. Turns out that's kind of hard to do. Now, we can do that if we have tree rings, because tree rings, the interior ring dies when the exterior ring is formed. So we know the year that tree ring died. And so we can say, okay, I can measure the carbon-14 that's in that tree ring now. I know how old it is. So that allows me to figure out how much carbon-14 should exist in these other things that were alive at the same time. So that's called a calibration. Problem is, the oldest tree ring that exists is about 4,800 years old. The oldest tree ring we've been able to test for carbon-14 is 3,500 years old. So that nice calibration runs out at 3,500 years ago. After that, they try other calibrations, but they get more and more absurd. So in the end, carbon-14 dating is really only a good tool for certain archaeological issues. It gets worse as you go farther back. And of course, the potassium-argon dating and things like that lead to millions of years, those have lots of problems. We'll stop after this, but the one thing I'll say is, if I ever have the opportunity to use two different radioactive dating methods, they almost always disagree. It's very rare that they will agree. So for example, every now and again you'll find an igneous rock, igneous rocks are made from lava, that's surrounding the remnants of a piece of wood. The wood can be carbon-14 dated because it had carbon in it. The igneous rock is dated with potassium-argon dating. And usually the potassium-argon date says 2 to 3 million years old. And the carbon-14 date says 2 to 3,000 years old. And this is the common result. The common result is if you have two independent methods, they almost never agree. And that's why I don't believe any of this radioactive dating stuff. And I know enough about radiation to know the problems associated with the assumptions. But mostly just from a flat out, when two different methods disagree by so much, it's probably both of them were wrong. This next talk's going to be very, very different. It's not going to be nearly as much science. It's going to be a lot of history and stuff. It's really fun.
Let's go ahead and say thank you to Dr. J. So we're going to take a 15-minute break and use the restrooms. There'll be snacks, and then we'll call you back in. And make sure you ask Dr. J about his current radioactive cat that he has. So let's go ahead and take a 15-minute break. We'll come back.